Alright guys, welcome back to Underground Science. In this video we're going to be talking about transcription, okay? So transcription is when you go from DNA to mRNA, okay? That's transcription, that's it. So if we look at this, right, let's start with talking about eukaryotes first. So in eukaryotes, there's three main types of RNA polymerases that we should focus on, okay? The first type of RNA polymerase is called RNA polymerase type 1, okay? RNA polymerase type 1 is good for making rRNA, or also called ribosomal RNA. So obviously, because of the name, right, rRNA is present in ribosomes, okay? rRNA is made in the nucleolus, okay? So ribosomal RNA is present in the nucleolus. Another fun fact, right, when I was trying to research about this was that sometimes in cancer cells, you can see the nucleolus hypertrophy. Okay, so you may see like some cancer cells have a larger than normal nucleolus. Okay, so I thought that was pretty interesting, that related to the nucleolus. Okay, main thing that we have to focus on is RNA polymerase 1 makes rRNA, which is present in the nucleolus. Okay, rRNA is present in the ribosomes. Okay, because it's called ribosomal RNA. All right. The next type of RNA polymerase we should focus on is called RNA polymerase type 2, okay? RNA polymerase type 2 makes mRNA, small nuclear RNA, and microRNA, okay? And then the last type is RNA polymerase type 3, and RNA polymerase type 3 makes tRNA and 5S rRNA, all right? So those are the three main types of RNA polymerases that we should know and what it makes, all right? So let's move on. Okay, so in prokaryotes, right, we should also focus on the RNA polymerase in prokaryotes. So you know how we just said eukaryotes, we just went through three, RNA polymerase 1, 2, and 3. Prokaryotes, they only have one RNA polymerase, right? So their RNA polymerase makes all the types of RNA, like mRNA, RNA, everything, right? But we have three different types. They only have one RNA polymerase that makes everything. Their RNA polymerase is made up of two things, a core portion, right, a core part and a sigma part. So specifically, it's a core enzyme and you have a sigma factor, okay? If you combine the core enzyme and the sigma factor, that makes up the RNA polymerase holoenzyme, okay? So that's what it's called. That's the RNA polymerase name for prokaryotes, okay? They have an RNA polymerase holoenzyme. It's just one RNA polymerase made up of a core enzyme part and a sigma factor. The core enzyme is made up of two alpha subunits, okay? A beta subunit, a beta prime subunit, and an omega subunit. Okay, that makes up the core enzyme. The sigma factor is on the side of the core enzyme, is attached, I should say, to the core enzyme, and it's helpful for binding to the promoter. Okay, but let's focus on eukaryotes after this. Okay, but it's just important that we should know that prokaryotes have one RNA polymerase enzyme. It's made up of an alpha or two alpha subunits, beta, beta prime, and omega subunit. Also has a sigma factor, and if you combine the core enzyme and sigma factor, you're going to get the RNA polymerase holoenzyme, okay? So let's dive a little deeper, all right? So if we look here, right, we have a coding strand, right? So we have a DNA strand that goes from 5' to 3'. So let's focus on the specific parts. So right here in the dotted red line is where the RNA polymerase will start transcribing, okay? It's going to start converting the DNA into RNA. So that's called the transcription start site. Before it, or behind it, I should say, or upstream, we have the promoter. Okay, that's where the RNA polymerase will bind to. All right. Then afterwards, right, so if we see this space right here, right, this space right here, right, between the transcription start site and between this white line, this is going to be the 5' untranslated region. Okay. Then between these two white lines, okay, from here all the way to here, is going to be the open reading frame. Okay, so that's going to be consisting of the exons and the introns. Okay, that's what's going to be. We're going to actually transcribe this and go on to translate it, but of course we'll see that we cut out the introns. Then from this white line and to the end, we're going to have the 3' untranslated region. This is where we're going to see the polyadenylation signal. Okay, and we'll go over that soon. All right, so again, between this transcription start site and the open reading frame, we have the 5' untranslated region, then we have the open reading frame, right, where this is the part that actually consists of the exons and the introns. The exons will go on to code or go on to the exons are mRNA, and then after that's going to turn into proteins, right, through translation. Then after the open reading frame to the end, we have the 3' untranslated region. So forgot about the prime part, right? And the 3' untranslated region is what's going to contain, right? The 3' untranslated region will contain the polyadenylation signal, okay, which will be important when we talk about processing, right? This pre-mRNA processing, okay? So that's that so far, right? Now let's look at more things about this, okay? So I tried to draw this a little big, but it may be a little bit crowded, but I think we can make it out well. So we saw that, again, we have the promoter, right? Specifically, though, we should be talking about the Tata box, okay? In eukaryotes, we can see many things that are present within the promoter that's going to help the RNA polymerase bind to it. So the RNA polymerase can bind to the Tata box. It can bind to something else called the GC box. It can bind to a CAT box, or specifically it's CAAT, right? So we have Tata, CAAT, and GC box, okay? These are all sequences, or these are all sequence of nucleotides that RNA polymerase can bind to, okay? But specifically, if we're focusing on the Tata box, this is located 25 to 30 base pairs upstream of the transcription start site. So if the transcription start site is there, the Tata box is located 25 nucleotides this way, okay? 25 to 30 nucleotides this way. That's where the RNA polymerase will bind, right, which is in orange. So that's the RNA polymerase will bind to the Tata box in this case, and then it will then do transcription, right? But first thing that we have to focus on is our transcription factors, okay? These are very important because what happens is, first thing what you're going to see is this Tata box where the RNA polymerase will bind, before the RNA polymerase binds, transcription factor 2D, okay, will bind to the Tata box. Because transcription factor 2D actually has a Tata binding domain, so that's why it can come here and bind. Now there's other transcription factors that we didn't list. We could have said we also have transcription factor 2E, A, H, right, and so on. But the main thing to focus on is transcription factor 2D because that's the one that has the Tata binding domain. And this helps RNA polymerase come over near the Tata box and bind, okay? And then transcription can follow after, right? So RNA polymerase 2 binds the promoter, and then we have transcription. But before that, first, transcription factor 2D will bind via its Tata binding domain to the Tata box. Then, second, RNA polymerase 2 will bind the promoter so that transcription can begin. All right, so let's see how this happens. So again, RNA polymerase came, right? And then it unwinds the DNA, and so it can begin transcription, right? So let's see how this transcription works. Okay, so remember DNA strand is made up of, or our two DNA strands. One of them is going to be the template strand, okay? One of them is going to be a template strand, which is going to be called the antisense strand. So it's going to be the strand that's going from 3' to 5'. And then we're going to have the coding strand, which is going to be called the sense strand. And that's going to be going from 5' all the way to 3' over here, right? So the top strand is the template strand. The bottom strand is the coding strand. In transcription, we're going to be transcribing the template strand, okay, which is the strand that goes from 3' to 5'. All right? So let's see how this works, right? So what we're doing here is we're taking this DNA, right, this template strand, and we're making RNA from it. So what did we say? Which type of RNA polymerase did we say in eukaryotes makes or converts DNA to RNA, right? Or which type of RNA polymerase makes mRNA from DNA, right? We said that was RNA polymerase type 2. So that's the type of RNA polymerase we'll be using. All right, so let's start. Let's start this transcription process, okay? So this is our RNA polymerase 2, and we're going to be going along the template strand, reading it from 3' to 5', and synthesizing our RNA strand or our mRNA strand from 5' to 3', okay? So let's go and see how this works. Let's go three nucleotides at a time, okay? So let's transcribe TAC. So what's the RNA complement to TAC, right? So let's do this in green, okay? So we're going to make our mRNA strand in green. So what's the RNA complement to TAC? That would be AUG, right? So let's go and write that down. So we would have AUG, right? So now the RNA polymerase has moved to right here. So let's go and change that color. We have RNA polymerase right here, and so far it's made what? AUG, right? So we have 5' here, and we're synthesizing towards 3' end. Now let's look at the next three. GAA. What's the RNA complement to GAA? That would be CUU, right? So let's go and write that in. So let's erase this, right? And then now we would have, let's go and change to green, right? We would have CUU, okay? So now our RNA polymerase is right here, right? So see, the RNA polymerase is moving along this way. Well, which way is this? This is from the 5' to the 3' end, right? It's reading from the 3' to 5', right? It's reading the DNA template strand from 3 to 5, but it's synthesizing its own mRNA strand from 5 to 3. Okay, so now we're over here. So we have AUG, CUU. Now let's look at the next three. That's TGA, right? So what's the RNA complement to TGA? That would be ACU, okay? So it's ACU. So let's go and write that in. So we have ACU, okay? So now the RNA polymerase is right here. Let's look at the next three, okay? We have TTT. What's the RNA equivalent or RNA complement to TTT? That would be AAA, right? So let's go and write that down. Okay, so we're going to have AAA. Now our RNA polymerase is right here. Okay, what's the RNA complement to TCG? That would be AGC. Okay, so let's go and write that down. So that would be what? AGC. Now the RNA polymerase is over here, right? What's the RNA complement to CCT? That would be GGA. Okay, let's go and write that down. So right now we have GGA. Okay, now the RNA polymerase is right here, right? We have 3 more to go basically, okay? What's the RNA complement to ATC? That would be UAG, okay? So let's go and write that down, right? That would be UAG. Okay, sweet. That's our mRNA strand, right? That we just made our RNA strand. This will go on. Well, first this has to get processed, and then it'll go on to make protein. This mRNA strand will go on to be translated into protein. But here's the thing, right? Remember, we recognize UAG, right? UAG is a stop codon. So once RNA polymerase 2 sees this is where it's going to end its transcription process. However, the reality is, right, what happens is after the stop codon, if you go on and see this coding strand, you'll have a 3' untranslated region, which we talked about in the previous slides. This region has, in the coding strand, it has AAT, AAA. So on the template strand, it would have TTA, TTT, right? Because that's what's complement to it. So what it's going to do is RNA polymerase 2 won't actually stop at the stop codon. It's going to keep on going, okay? It's going to keep on going. So let's see what that would look like, and we'll know why in a sec. So if it keeps on going, right, it's going to translate this, or it's going to transcribe this into A, U, A, right? So it actually ends here, right here, around right here, because this is the 3' untranslated region. This won't go on to make protein because it's called the 3' untranslated region. It will not get translated. But you need RNA polymerase 2 to continue on. Even after the stop codon, it has to continue on and translate this sequence into this, right? Because this right here is very important. This is called the AAU-AAA, is the polyadenylation signal, okay? The polyadenylation signal. That's going to be very important when we talk about processing and adding a bunch of adenines to this, okay, in the next video. So we make the polyadenylation signal, or we transcribe the polyadenylation signal, and then we get AAU, AAA. All right. Then we don't have to go into detail about this, but what happens is actually an endonuclease would come in and try to cleave like a few, like let's say around 20 nucleotides this way, right, of where RNA polymerase 2 stopped, because like technically RNA polymerase 2 could go a little bit further until around 20 nucleotides, and then endonucleases would come and cleave it. But for now, the main thing to understand is RNA polymerase 2 cannot stop at the stop codon. It has to go a little further into the 3' untranslated region, because it needs to put down this AAU, AAA, because this is the polyadenylation signal, okay? So yeah, that's basically it. So what we end up as, right, is this. This is what we end up as. So remember we said that we have the 5', then we have the 5' end of the mRNA strand, then the 5' untranslated region, then we just transcribed this, right? This is our transcribed pre-mRNA. Then we have our 3' untranslated region where we had that AAU, AAA, right? Because we just transcribed that. RNA polymerase 2 went into this 3' untranslated region and transcribed the template DNA strand, and we got AAU, AAA. This is the polyadenylation signal. Okay, in the next video, we're going to go over processing of pre-mRNA and splicing of pre-mRNA. All right, so I hope to see you guys there. Thanks for watching.